welcome to our Biognosys breakfast seminar. I'm glad to see a pretty full hall right here. I think for anyone that knows Biognosys, we've been hosting breakfast seminar for Spectronaut, one of our flagship software, for over 10 years. But this marks definitely the inaugural breakfast seminar that is focused more on our contract research services. And it's definitely a pleasure to have our guest speakers here from the biopharma industry, and also great to have you as a great audience so early in the morning. So I want to give a very quick introduction before moving to the introduction to our speakers. And I think this is one of the slides where everyone has seen in different formats for probably hundreds or thousands of times. So the takeaway is very easy, right? I mean, it takes a lot of time, or it takes a village and a lot of cash to develop a drug. And the question we would like to support with mass spec-based proteomics is how we can accelerate or de-risk the process at different stages. So really from starting early discovery up to preclinical into clinical trials. And this is also the topics that we want to cover today. Of course, we cannot talk about everything in 1 hour, but we really want to focus on some of the most expensive part in drug discovery, namely how you move a molecule actually from preclinical into IND into early phase 1 clinical trial, and then another talk focusing on how can you leverage biomarker in the late clinical trial, for example, for patient selection, stratification, et cetera, to make sure that the trial can be run with a higher success probability. Last but not least, we will also present you some of the newer approaches that we've recently developed for plasma proteomics. So please stay until the end. Hopefully there's enough coffee to keep one awake, and also hopefully the content will be exciting as the breakfast. With that, I would like to introduce the 4 speakers we'll have for today's seminar, starting with Eric Khun from Kymera Therapeutics, who will walk us through the newest, most exciting developments of targeted protein degrader, one of the most exciting modality now in the drug discovery field, and how mass spectrometry is a great fit along the entire development pipeline from really early discovery up into the clinic. And the second talk is something I think everyone in the mass spec field is very aware of. Can an assay be transferred from lab to a lab? So in this case, what we're doing is to transfer a complicated hybrid targeted mass spectrometry assay across the pond from Cambridge, Massachusetts, to Schlieren in Switzerland, and to see how this assay performs and can this actually be automated and applied. Last but not least, I know you see a different picture in our advertisement and on the board outside, where our CEO Oliver is supposed to present the newest plasma workflow that we've developed at Biognosys. But we decided that no one is actually better than Roland, who actually worked hands-on on it, so he can also better answer all the questions. So we made this last-minute adjustment, more dedicated to the science. With that, I would like to introduce our first guest speaker, Eric Khun from Kymera Therapeutics. So Eric is Associate Director for Targeted Proteomics at Kymera. I think people who have been in mass spec field for a while certainly knows Eric's name from different papers. So Eric has spent more than 16 years at the Broad Institute developing assays for multiple large collaborations and consortium, for example, CPTAC. And before that, Eric has spent more than 20 years in the pharmaceutical industry, being responsible for analytical development and biomarker discovery. So without further ado, I'd like to hand it over to Eric, and wish everyone enjoy breakfast and a great presentation.
Wonderful.
Thank you. I'll change it down.
Thank you, Johan. Good morning, everyone. Thank you for coming out early on a Monday morning. Very excited to share. But first, I'd like to just thank Biognosys in general, Jakob, Johan, Oliver, Roland, for the invitation and the opportunity to talk to you today about advances in approaches for using target protein mass spectrometry, target protein mass spec, target mass spec, to measure pharmacodynamic effects of target protein degradation in the clinic. So just a quick disclaimer. I'll start just by introducing the TPD, the target protein modality, how at Kymera, quantitative proteomics has really enabled the development of drug TPD degraders throughout the discovery and drug discovery pipeline, highlighting, oops, which button we have here? Highlighting a paper that came out late last year in Nature Medicine where we were able to show in patients with atopic dermatitis and hidradenitis suppurativa, deep and sustained degradation of IRAK4 in PBMCs from blood and skin using targeted mass spec. And before that, really going back into some of the pre-analytical, assay development that was, the advantage of a degrader versus an inhibitor. Other particular focuses on sample preparation so that everything in the assay was ready to go before that first in-human study. And then finally, there are conclusions, primarily that the assay is fit for purpose to do exploratory measurements of target protein degradation, and that we're still working on challenges such as PBMC stability, so we can really include multi-center trials and dealing with recovery and methods for extraction from tissues that are very heterogeneous, like skin. So at Kymera, we're really inventing new medicines, harnessing the power of the ubiquitin proteasome system. The ubiquitin proteasome recycles, removes unfolded, denatured, aged proteins using enzymatic cleavages that it sort of gets, the indicator there, recycled back. In TPD, we're taking a small molecule that binds to the protein of interest, tethering it together close to an E3 ligase in the presence of an E2 ligase that allows ubiquitin tags to be placed on that protein, and as it's earmarked, gets shuttled then into the proteasome and degraded. And this process is actually catalytic. This degrader can go back and repeat the process over and over in the cell. And so this functionality actually opens up more of the proteome to be drugged. Compared for some of our molecules, IRAK4, MDM2, TYK2 inhibitors don't get full inhibition or don't sustain the inhibition for periods of time. And with a heterobifunctional degrader, you can actually remove the protein altogether. In other cases, there are undrugged targets, STAT3, STAT6, that the pocket is not accessible for inhibition. And there you can just also remove the protein altogether, either with a bifunctional degrader or also with molecular glues. And then finally, there's a lot of protein expression that's varied across tissues. And so picking a different E3 may actually enable to target different tissues or remove the liabilities associated with a pan-drug therapy where toxicity may really prevent it from being used in the clinic. And so at Kymera, we have two molecules that are in phase 1 studies for oncology. We have STAT3 in liquid and solid tumors and an MDM2 degrader for other indications. We also have two molecules that are about to enter phase 1s, one for STAT6 and one for TYK2 with multiple different indications. Today, I'm just going to really focus on our lead molecule, KT-474, an IRAK4 degrader that we have developed in partnership with Sanofi. And so with the continued advancement of proteomics technologies, the instrumentation, the software, the hardware, the computational tools, and so on, that increase the depth and breadth of the proteome that can be measured and quantified by mass spectrometry, including post-translational modifications, medicinal chemists continue to develop drugs to remove, modulate, and change the levels of one protein. And so there is still a great opportunity to have assays that can measure one or two proteins with really deep, sensitive, and reproducible measurements. Biomarker assays, whether it's proteomics or in other assays in general, are typically expected to measure proteins that go up in concentration. In targeted protein degradation, we're measuring things that decrease by 90% or greater. And so assays that can be linear down to 1 order lower or 2 in cases where the protein is in much lower concentration of the cells really are an advantage. In addition, the clinical timelines, sorry, let me take a step back. I mean, there are multiple enrichment strategies to improve the sensitivity of detection, whether it's antibody enrichment or fractionation or other ways, but those take time to develop and qualify. And so looking for a method that can directly measure the protein of interest on a nanoscale that can be transferred to CROs supporting the clinic is really imperative to keep the assay relevant relative to PK and the other assays such as ELISA on that clinical timescale. And so here is an overview of how we use quantitative proteomics at Kymera to qualify and develop drugs and degraders. We really start with a discovery experiment, whether it's DIA, TMT, offline fractionation, deep dive, thousands, 8,000, 9,000, 10,000 proteins where we're looking for potential off-targets, as well as potential biomarkers. From there, we will take those peptides and proteins and develop stable isotope labeled peptides, LC-PRM, and direct measurement assays that will allow us to move into in vivo studies in mouse, rat, dog, monkey, rabbit, pig, where we have peptides specific for each species, where we can then measure the degradation in cells and also in tissues, heart, liver, kidney, lung, spleen, and especially skin, which is really a challenge. And eventually those assays can then be translated and made robust and qualified for use in the clinic, which is what we're doing right now for STAT6 and TYK2, getting ready for those first-in-human studies. So our first-in-class degrader, I'll delve more into degrading IRAK4 and the implications for biology in the clinic. IRAK4 is sort of a master regulator of innate immunity. You know, tethered with MyD88, it will take input from toll-like receptor agonists as well as interleukin-1 receptor cytokine input and start the either in a catalytic function or in scaffolding pro-inflammatory inflammation, Th1/2/17 immune response. And compared to an inhibitor, shown here with a readout of interleukin-6 after stimulation, the degrader is more potent, it's much more left-shifted, and it's a more sustained reduction of that cytokine blocking inflammation, really showing that there is an advantage to removing the protein altogether. In addition, we've done phosphoproteome-wide studies to show the reduction of downstream kinase activity and verified this more in efficacy models, in preclinical efficacy models. And so assays were developed to measure IRAK4, both a flow assay shown here on the left and antibody-based, where you can measure IRAK4 degradation across a number of cell types, as well as calibration curves shown here in PBMCs at Biognosys, linear range, getting back to that aforementioned linear range of 1 or 2 orders deep degradation we can measure with these assays. The antibody assay, in contrast, has got this background noise that really prevents measuring really sustained based on the noise of the antibodies and the fluorescence readout in the system. But also collecting patient blood over periods of time and intervals, there are limits to how much you can draw and when you can draw it and how you're going to process it. And so we developed a method that allows us to get from a single draw of blood both plasma for the PK and small molecule quantitation and PBMCs for PD with targeted mass spec. We also measured with both peptides sort of the relative stability in the first 4 hours, which is the range in which most of this isolation would take place. And then looking at in a dose curve in PBMCs with this degrader, you can see single-digit nanomolar degradation, a very potent IRAK4 degradation by both peptides. And then also showing similar levels of quant, whether we're doing it in skin, which is much lower than in PBMCs. This is sort of just an approach of the study that was used in healthy volunteers, showing both proof of mechanism by flow and mass spec, as well as biology with an ex vivo stimulation measuring the cytokines in plasma. On the other side, skin was also collected for similar proof. IF was done, staining to show degradation of IRAK4, mass spec, and then RNA-seq, which I'm not going to present here today. The trial design itself, there was 2 phases. The first, parts A and B, A being a single dose study with a number of cohorts, and then also a multiple ascending dose with daily dosing out to 14 days across a number of doses shown there. Part C was really the part of the second phase of the study, went out to 28 open label, 28 daily doses. It was open labeling in patients with HS and AD. And of course, with all phase 1 studies, the primary readout is safety and tolerability. But it's also an opportunity as an exploratory to measure the knockdown of IRAK4 and PBMCs in the skin. Shown here is figure 1 for that Nature Medicine paper. On the left is the absolute quantitation of femtomole per microgram IRAK4, coming down to a nadir around 7 days for most all doses. And that's very close to the limits of quantitation here in the assay, and it continued out to day 14. This is in the MAD portion of the study, the 14-day daily dosing. And then you see sort of a dose-dependent rebound after out to 28 days. On the right is how we plot a lot of these data relative to the pre-dose, showing deep degradation greater than 90%, greater than 95%, both at day 7 and day 14 for all the doses in the study that were statistically significant there. But also in skin, we were able to get good exposure from an oral dose degrader, much higher than we saw in plasma. And based on this dosing, if you compare, this is for AD and HS, the pre-dose compared to the levels of IRAK4 measured by targeted mass spectrometry, they return to be similar to those healthy volunteers in the MAD study. So that indicates that we're engaging the target, we're reducing it back to levels, and we're actually seeing a decrease in those elevated levels in those 2 indications. So in conclusion, between the SAD and MAD studies, between the oral dosing, there was deep, robust, sustained degradation of IRAK4, both in the single and the multiple dose studies. It was well tolerated. And then in the patient cohorts, we saw similar degradation, and we even saw some very promising preclinical activity based on this. And based on this study and based on these results, the assay and the phase 2 study was then transferred to Sanofi, and that's going on right now. And this is just more indication of sort of a reduction here in the HS patients in addition to the degradation. But that's not the only project we're working on. We also have an Iraqamid where we've been able to decrease IRAK4 by mass spec, and Icaros and Ilos, STAT3 degradation for our STAT3 program. And we also have other assays for the MDM2 program as well to show a pharmacodynamic effect of those degraders. So we're very encouraged, very excited about all this progress. In short, the assay is fit for purpose for measuring that, but it was really based on all the background, the foundation, and the framework that was developed before we got to the clinic, whether that was just the cross-species validation, how we handled the samples so that the assay was ready, and then we also had mechanisms in place to move this over and translate the assay across sites. There's still challenges that we are pursuing. PBM stability, that 0 to 4 hours, we would like to extend that. That'll allow us to process more samples, bring in multi-centers, have a bigger, expansive trial. And then, as I alluded to earlier, getting more reproducible extraction efficiency of our targets out of a very heterogeneous tissue such as skin, especially a skin that's being divided in many ways, just like the plasma and the blood draws for different readouts, so we can use all these together. And hopefully I've shown you today that we're really using target mass spectrometry as a suite of assays, whether it's flow, IHC, ELISA, to really comprehensively understand the pharmacodynamic effects of our degraders. And with that, I would like to thank you for your attention. Happy to answer any questions. One of the answers to this question is it does get sunny in Boston, just not today. So the sun does shine, but not on the day we had our summer outing. Thank you. Yes. That's a great question. As an exploratory assay, we looked at some stability, we looked at calibration curves. We did not do a full validation in the sense that this is an endpoint in any trial. In fact, as you go to phase 2 trials, they'll probably not likely use a lot of this readout. They've already shown that they can get degradation. So we're really qualifying these assays with stability, reproducibility, precision, sensitivity, and more in that line as a qualification for an exploratory readout, not the final endpoint within the trials themselves. Thanks. Thank you. Yes? Yeah, I think there's still a niche. That's a great question. We're still in the process of using DIA a lot for our in vitro studies, in the selectivity and off-targets to verify those DC50s and Dmax by DIA, and can we get the same result by PRM? I think translating a DIA method into the clinic is going to be that's a longer road to hoe, where right now we're going to still do that transition so we can get 20 scans under the peak, more precise quantitation, and have a number. This number, percent degradation, I can go to it too much. But a lot of times they don't have an efficacy readout, cytokine or other. So showing you can reach 90% degradation or greater is actually going to be an efficacy readout. And so having more precision around that number allows the doctors and everyone to make a call on how well we're engaging the target. So yeah, DIA has come along and helped us a lot with the selection of peptides for targets and building the assay, but eventually it has to transfer into an in vivo setting, complex tissues, PBMCs, where we need that rigor, and I think that will continue as we move forward with those assays in the clinic. Yeah.
Yep. Thank you.
Yep, thanks.
Thank you, Eric. We will have a dedicated Q&A session at the end. Hopefully we'll have probably 10 minutes time, so you guys can still ask questions to Eric or any other of our speakers. So in the interest of time, let's move further to our next topic. Perfect. So this really showcased, a great scientific collaboration between us and the Biogen team. And Alan, who is a principal associate scientist at Biogen, who has more than 10 years experience in fluid biomarker development, especially focusing on highly sensitive assays on low abundant proteins. So Alan will start and tell us more about how they develop a hybrid targeted assay for LRRK2 at Biogen, and then will be followed by our senior scientist Luca, who will talk about how we transfer these assays over the pond to Switzerland. So welcome, Alan.
Hi. Testing. There we go. Hi, I'm Alan Shomo. First, I want to thank the Biognosys team for inviting me here and to talk about exciting collaboration that we've had together. Okay. Down. So today, I'm going to be talking about the internal development of a quantitative CSF assay for the LRRK2 protein and how we are deploying this in clinical studies. And this has been a large collaboration of the pretty much the whole Biogen biomarkers department. So on a high-level overview of LRRK2 and Parkinson's disease, Parkinson's is the second most common neurodegenerative disease with over 1 million affected in the US. It is characterized by dopamine-containing neurons in the midbrain, which become susceptible to degeneration, which leads to motor dysfunction. LRRK2 is a large multi-domain protein with several different activities, including GTPase activity and kinase activity. And it is involved in vesicle trafficking and lysosomal function. And the mutations that are disease affected are actually gain of function and are the largest genetic risk factor for late onset PD, which makes it an attractive therapeutic target, of which there are several programs. Most of these therapies are kind of designed to either lower the entire expression of the protein or its activity. Two such studies that are currently in clinical studies are LRRK2 ASO between Biogen and Ionis collaboration, and a few different programs for small molecule inhibitors. One, a collaboration between Biogen and Denali, and then a separate one run by a Neuron 23. Now, in order to support these ongoing clinical studies, a CSF measurement of this protein to be a surrogate to the actual CNS compartment was desired. So the Biogen biomarkers team started with developing two very sensitive immunoassays to target total LRRK2, and these performed extremely well in tissues, primarily including primate brain, rodent brain, and human PBMCs. Unfortunately, even after troubleshooting and method development, results were more inconsistent in human CSF, with over half the samples tested falling below the limit of quantitation using ELISA. And reasons for this inconsistency have been hypothesized, usually around the capture antibody not being able to reach the epitope for what reasons, possibly truncations, misfolding, heterodimerizations, etc. So in order to fill this gap and make a functional CSF assay, we went with a hybrid LCMS approach. And we have two kind of types of hybrid LCMS approaches that we use in our lab. One being anti-protein pull down, in which we use an anti-protein antibody to pull down the entire protein out of the sample, do a trypsin digestion, and then spike in a isotopically labeled version of the target peptide we're interested in, and then run it through a targeted LCMS, usually PRM. And we are using, obviously, the peak ratio of the light to heavy to quantitate. And the advantage of doing this is there's usually a large variety of anti-protein antibodies available that we can just buy off the shelf, screen, and get started with method development. But the disadvantages are, we are injecting a trypsin digest, which has some complexity to the sample, which can limit signal to noise. To kind of maximize the sensitivity that we needed for this assay, we went with the cis-GAPA approach, where we digested up front the whole sample with trypsin, spiked in our isotopically labeled peptide, and used a specific anti-peptide antibody to pull down both of these targets. This leads to a very enriched and clean sample, which can maximize the signal to noise. Obviously, the disadvantage of this, which makes it not possible for every assay, is you need a good specific antibody just for that peptide. Fortunately, the Michael J. Fox Foundation had sponsored a LRRK2 epitope mapping project, which we were able to use the results from to pick a couple promising target peptides. The highlighted ones are the two that we chose. The N241 was a clear winner, so that's the one we will be talking about throughout the rest of the talk. One of the first experiments we did to characterize this antibody was to do a data dependence DDA kind of experiment on the trypsin pull down to kind of get an idea, characterize what the antibody is actually pulling down. We expected primarily the fully cleaved peptide, and we saw it, but we also noticed that the single miscleave peptide was giving us about 10-fold more signal. Now, this is an interesting miscleavage peptide. There's a double lysine on the N-terminus, and even though miscleavage peptides, we tend to not go to those first because they can be sensitive to digestion conditions. Literature has kind of shown that in this double lysine scenario, the miscleavage is actually favored kinetically. So we decided to try to use this to get the sensitivity boost that we needed and to further characterize it. After some early method development, we decided to do a correlational study with the current ELISA that we had in-house. And we did this in tissue, cynocortex, that was treated with LRRK2 ASO. And as you can see with LCMS in red and the ELISA in blue, that they correlated very well with each other, which gave us a lot of confidence that we picked a correct surrogate peptide for this assay. And that this miscleavage is not affecting the assay so far. So we further did a proof of fit for purpose, like partial analytical validation. We looked at precision. We looked at a lot of things. We demonstrated that in recombinant, this peptide gives us very good reproducible linearity. And most importantly, in human CSF, we were able to quantitate every sample that we tested very precisely, which really gave us confidence that we picked this kind of odd miscleaved peptide really gave us the sensitivity boost we needed to make this assay possible. So then we continued to run actual human studies. We decided to run first the LCC sample set from the Michael J. Fox Foundation. We ran 135 samples in a blinded fashion. And this sample set is divided into 4 different groups. One is a kind of control group. Then we have a idiopathic Parkinson's disease group without the LRRK2 mutation, LRRK2 carriers without symptomatic Parkinson's, and verified LRRK2 Parkinson's patients. And we were able to show that using this assay, a separation between both the LRRK2 carriers and non-LRRK2 carriers and with disease status. And it's always really exciting to see when you, after the long development of an assay, that you actually see a clear biological effect. And this gave us kind of the confidence we needed that this would, we can use this to run clinical samples and inform on the biology. And just as a reminder, the LRRK2 mutations that we are looking at are gain of function, therefore the therapeutics are either lowering the expression or inhibiting the activity. And we're measuring this in cerebral spinal fluid as kind of a close to the actual compartment that the drug is being treated in, and it's also accessible for clinical samples. And we deployed this assay in a phase 1 study for one of the for the kinase inhibitor drug, and using this assay we were able to show a dose-dependent lowering of total LRRK2, a pharmacodynamic effect, which was also really exciting. And at this point we started reaching out to external partners to see, number one, to find one that had the technology and the expertise to replicate this this assay, but also the experience to increase the throughput and the regulatory experience needed to progress to later phase trials. So in conclusion, Biogen had developed several immunoassays that were very sensitive. It worked well in tissues, unfortunately not CSF. We filled this gap by developing a mass spec method, SYSCAPA method, which was really made possible by the Michael J. Fox Foundation's work. And we used that data to discover a very sensitive, highly charged peptide that really made the CSF detection possible for this assay. And using it, we were able to distinguish between genotype, age, and Parkinson's status using LRRK2 measurements. And this assay is continuing to be deployed for our LRRK2 therapeutic studies. We showed a phase 1 study with the kinase inhibitor showing a pharmacodynamic response with LRRK2 protein. Unfortunately, early versions of this assay were tube-based, very hands-on, high effort, and low throughput. And we really wanted to reach out to start externalizing people with experience to not only transfer this assay, but also with experience scaling up and running in a regulated environment. So I would like to thank Biognosys for the invitation and the whole Biogen biomarker team, including Omar Babrook, which led most of this work, and John And, which led the transfer. And with this, I will pass to Luca to talk about the tech transfer. Thank you.
Hello, hello. Okay. Thank you so much, Alan, for this very nice presentation. My name is Luca, and I'm a senior scientist in Biognosys R&D wet lab team. And for the second part of this presentation, as already announced, I would like to focus on how we could actually transfer this assay between the two organizations from Biogen to Biognosys. So when Biogen approached us that they actually want to outsource this assay, we were immediately really motivated to work on this. So we settled on a few key requirements on the workflow. Of course, our Biogen had some specific needs in terms of sensitivity of the assay and reproducibility. They wanted to have a scalable workflow and also an efficient one. So how could we achieve this? So we needed to discuss a few key aspects of the workflow, mainly concerning the sample preparation and then also the LCMS acquisition. So if we look at the sample preparation, as already introduced by Alan, they have this published LARC2 this kappa assay, and in the meantime, they also worked on an internal semi-automated protocol. On the Biognosys side, we have a fully automated sample preparation workstation with a predefined hardware setup. So here it was really key to actually transfer from the different hardware setups the sample preparation. On the LCMS acquisition side, it was a bit easier. It was Biogen is using an LSF1 and an Exploris 480 as a mass spectrometer, and these are both devices we have in-house available. So this led us to define 4 main work packages for the project execution. So first of all, we wanted to have the LCMS setup dialed and reach the sensitivity for this target peptides. Once this is working, we focused on the most time-consuming step, which was establishing the sample preparation. So we wanted to have an automated sample preparation here. Then we would run an assay proof of principle and do some optimizations if necessary. Then as a third part, it was to actually combine the first two and work on the sensitivity and the reproducibility of the assay. And last but not least, it was to quantify some real biological samples with a small CSF cohort. So let's focus on the first part. So we were using here a PRM, or a parallel reaction monitoring assay. Just a quick reminder, so how it works. You would have the peptides eluting down from your LC system, which are then ionized. A precursor ion is selected, fragmented, and then separated according to its mass-to-charge ratio. A spectra is generated, and then we can run the data analysis, quantify the peptides, and then also in the end infer back to the protein. So again, for this, we used the EVOSEP1 as predefined by Biogen. We used the Whisper method, so the high sensitivity methods, because these peptides are really low in abundance, and we work with the 40 samples per day throughput method. Then on the mass spectrometry side, we used the Exploris 480 as also predefined by Biogen, and for the analysis, we use SpectraDive 11, our in-house software for targeted analysis. So this method has been established and fine-tuned on several different parameters to actually reach the target sensitivity. And this you can see here. So we did a dilution experiment where we just diluted down the target peptide, and on the x-axis you would have the concentration, on the y-axis the intensity, and we had this predefined limit of detection provided by Biogen. And when we assessed our method, we could nicely see that we can actually have a linear response down to our LOD. And if we look at some of the signals here for the highest ones, or here at the LOD, and then especially in comparison also to our blank value. So we could check the first box and move on to actually work on the sample preparation. So here again, it was essentially in the beginning to assess the hardware capabilities. So as said before, we had this manual protocol from Biogen, from their publication, and they also have this semi-automated protocol on an HLM Bravo system here. And on our end, we have our sample preparation workstation, which is mainly here a liquid handler connected to several third-party devices, and this we use for several different assays. For this assay, relevant third-party devices include, for example, thermoshakers, sealer peeler to perform incubations, and then we also use, of course, a magnet since we're working here with magnetic beads. So it was then time to actually implement this workflow, which I would, here as a reminder for the SysCap LRC2, like we would have 5 main steps. So we'd have some kind of preparation of the samples, but also the labor and the reagents. Then we perform the digestion of the CSF and also the spiking. In the meantime, we can in parallel couple the beads to the antibody, then perform the pull down of the target peptides, the light and the heavy, elute these and load them on the evo tips. And this was implemented, as I said, on the sample preparation workstation, and we had several development steps. So first we needed to come up with a concept to actually plan all the liquid handling steps and also the processing steps. Then I wrote a script to actually run it on the robot, which has then been tested. So we usually do first some logic testing if the logic works. Then we use some water runs where we only use water, and then some real reagents which need to be optimized so that we have the liquid handling dialed. So for example, that we don't have any contamination between the wells, or often we also need to reduce foaming and these kind of things. So the final workflow in the end for the end user is quite simple. So you would provide the samples, which can be loaded in a format of 1 to 96 samples in a 96 well plate, obviously. Then you would load the reagents and the labor and provide some input file with the sample details. You would run the processing, which is fully hands-off, and then you have, as an output, you get your samples again, 96 well plate. You would have a log file, so we have full traceability along the full workflow. So we know at each point what happens to each of the samples, and we also have a recording system installed, so we could always see and go back to the recording to actually see what happens to the sample. In terms of throughput, so if you would like to run a full 96 well plate, so the user would need to first prepare the reagents and the samples, then you would run for 7 to 8 hours, and then you would also in the end need to load the samples on the evo tip. And this workflow is easily scalable, so if needed, we could also run, for example, 2 plates in a day. So after setting up this method, it was time to actually do a biochemical proof of principle. So for this, we used a recombinant LRC2 protein, and then ran the full pipeline and did a PRM acquisition. And we did this just with 2 replicates, and the result we would see from the PRM acquisition was just what you see down here for the 3 replicates. For all of them, we have a really nice target from the light peptide, so from the endogenous peptide, and also we have a nice signal from the reference peptide from the spike-in. So which we could conclude that actually the pull-down or the biochemistry of the assay works. So we could also check the second box and then move on and actually combine the 2. So for this, we did a calibration curve. So we diluted down here the recombinant protein from 100 picogram per ml down to 0 to assess actually the sensitivity of the assay and also look at the reproducibility. And again, we had here a predefined limit of detection provided by Biogen, which was at 1 picogram per ml, which is quite low actually. And so on the x-axis, you would see concentration, and here the response from the peptide. And what we would see is a very nice linear response with quite a high coefficient of determination. And also, if we look at CVs for the replicates here, these are below 10%, and this is actually sample preparation and LCMS acquisition combined. Again, if we look at some of the signals here for the highest one, then for here 25 picograms per ml. And here, even like if we zoom in a bit and look at the values below 10 picogram per ml, we still have this nice linear correlation. And here at the lowest one for the LOD at 1 picogram per ml, we still get a good signal here. And here for the blank value. So with this, we were actually happy that the combination of the sample preparation and the LCMS acquisition works well. So we could move on to the final step and quantify some biological samples. And for this, we used 10 CSF samples, which were provided by Biogen, and they have been pre-assessed in their laboratory, and they were like values between 2 to 10 picograms per ml of the LRC2. And when we measured the same samples in-house, we were really happy to actually see that we almost exactly match these absolute quantities with a really high Pearson correlation up here. And here I really want to emphasize these are two different laboratories on two different continents, two different hardware setups for the sample preparation, and two different LCMS acquisitions. And we see almost similar results, which was great to see for us, and I think also Biogen was really happy to see this. So with this, I'm already at the end for my presentation. Just to conclude, I think we could successfully transfer this assay from Biogen to Biognosys. We now have reproducible, efficient, and scalable workflow. And moving on, we actually are working now on extending this assay to more targets. Then we do proper assay validation, and then the end goal would be to then measure RORC2 levels in clinical trials. And with this, I would just like to acknowledge some members of our Biognosys team from different departments, and especially also Biogen for trusting in us and working with us. Okay.
Thank you, Luca, for the great presentation. Of course, thank you, Alan. For our last talk, we'll have switched the topic a little bit to discovery. So I think you've seen in the previous two presentations, right, how mass spectrometry can deliver precise data in a really tight time frame, and how we can also reproduce data across different continents. Here we want to show something new, and I will not disclose too much. Roland, the stage is yours.
Hello, everybody. Good morning. So I'm Roland. I'm the head of R&D for ASMS at Biognosys, and I'm happy to present you the P2 plasma enrichment system that we have developed for discovery in plasma. Yeah. Blood is the window into the body. If it could pick up all the molecular clues that are in the body, we could actually measure what's going on in the different parts of the body. But blood is a difficult matrix. It poses a lot of analytical challenges, especially to mass spectrometry. So the dynamic range of the proteins is the most important challenge that we have in plasma. And if we just measure neat plasma, we are barely scratching the surface. So we get at best surrogate markers and not markers for the real biological processes and diseases. So the most widely used approach in the past to get deeper into plasma was using depletion. The idea there is to move the most abundant proteins and so shift down the dynamic range window. This works to some extent, but only gives us middle depth. So we are not able to reach down to the cytokines, to many of the proteins. It also comes with lower throughput and changes in reproducibility due to the immunodepletion. Then in the late 2000s, the opposite approach was presented by Pierre Righetti. It was based on enrichment of plasma proteins and de-enrichment of de-enrichment of high bound proteins and enrichment of lower bound proteins. And this is also the physical principle that our P2 system uses. So a strong de-enrichment of high bound proteins and enrichment of lower bound proteins. And we also, importantly, this comes not just for identification, but also without distorting quantification. So let's take 2 minutes quickly to look into the science behind it. For this, we need to look outside of proteomics. We need to look into nanomedicine. There it was first reported that particles that are designed to deliver drugs become coated with protein corona. This protein corona consists of, this protein coating consists of hard and a soft corona. And the soft corona is based on protein-protein interactions. It forms slowly and is less stable. So this is rather problematic for nanomedicine, but it turned out to be a very powerful principle for protein enrichment in proteomics for biomarker discovery. So what you see here, this depiction of the protein corona is rather inaccurate. And so let's take a look how this looks in reality. So this first EM picture shows a particle coated with a hard corona after intense washing. And the proteins are interacting directly with the particle surface in a small layer. So, and the second picture shows a full soft corona. It looks really different. So the proteins form a cotton candy-like structure. It's very extended. Clearly the proteins are far away from the particle, and they interact rather with each other than with the particle. So why are the proteins doing that? Why are they start to aggregate with each other when they are soluble in plasma alone, when they are exposed to a particle? There are several studies that report that the components of the unspecific immune system are involved. So it may well be that this resembles a phenomenon known from behavioral science, where the Asian bees form a bee corona around the Asian giant hornet and thereby neutralize it. So what can we learn from these bees? Our hypothesis that the protein corona is an active process, that if we can control it well, we can use it for plasma protein enrichment. Based on this principle, we set out to build an optimized particle enrichment system focusing on four main objectives. This: find the particle surface, topology, and buffer conditions that optimize the corona formation, then stabilize the soft corona for high reproducibility and quantitative precision, achieve an enrichment with a single well particle workflow, and achieve a throughput of at least 40 samples per day to enable large projects, and do all this at the cost structure that allows population-sized screens. We achieved all these objectives and are excited to make our P2 enrichment system accessible to the proteomics community. With an enrichment factor of 10-fold over neat plasma, it is to our knowledge the highest reported enrichment factor, which is a good benchmark of how the enrichment works. But what is even more important is then just the absolute numbers. It is very, very reproducible. There's a very low coefficient of variation. With P2, over 80% of the proteins have a CV lower than 20%. The median is also well below 10%. So again, this is better than methods that we have evaluated that are existing in the market so far. So this is likely thanks to the robust single well workflow that we are using and the optimized digestion and cleanup and washing conditions that successfully stabilize the labile soft corona. But you don't want just to enrich proteins, we also want to retain the quantitative information. So in the two species plasma mixture experiment, we see no sign of ratio compression. The measured fold changes in neat and in P2 are close to the expected fold change, which is the blue dotted line here. So the original proteominer plasma enrichment technology, it was explained that plasma enrichment would perform a plasma protein normalization, but we don't see this normalization or compression effect, which is very good news for quantification for biomarker discovery. So then what kind of proteins are we enriching for? So we see a very significant increase of relevant proteins of geoturns compared, for example, to NEAT or to the established MAC-resin-Sachs protocol magnet across all gene ontologies categories that we have visualized here, for example, by selected protein classes. This is specifically also true for membrane proteins and exosomal proteins that we looked at. What again is important characteristic is that P2 covers the proteome of other enrichment particles, including NEAT or also magnet resin Sachs depicted here. So all the proteins are contained, it's just getting deeper. This is good news because it means that users don't have to use different enrichment methods in parallel. So then what is the gain for biological information with the increased depth? To investigate this, we repeated our published study with four cancer cohort that consisted of lung, pancreatic cancer, and colorectal cancer, and matching healthy. And we prepared that data again, those samples again with P2, and also looked at the original data, and then we're reanalyzing them with the same classification algorithm to distinguish whether we can find out from plasma to which cancer type they belong. And doing that with the P2, this led to a dramatic jump in prediction accuracy of the cancer types from 70 to 90%. So we can assume that this can be explained by the higher number of tissue-secreted proteins that are more direct, that have a more direct relationship to the discriminating processes in the different cancer types. So with that, I come to the summary and to our highlights. So we present here our new P2 system, the plasma enrichment technology, which is a single well, high throughput enrichment method for highest depth and reproducibility. It can reach over 7,000 proteins in clinical studies in high throughput mode. And it will be available via our contract research services and via licensing and tech transfer for industry. And also additionally, importantly, it can be combined with the Alamar Biosciences Nulisa panels at Biognosys. And with that, I'm done. I thank you for your attention, and I wish you an exciting conference.